Climate change is no longer a distant threat. It's here, it's now, and it's real. The United Nations recognizes the urgent need for action through its Sustainability Development Goals, especially Goal 13, which calls for immediate efforts to combat climate change and its impacts. In India, the effects are undeniable. Between 2000 and 2021, deaths due to extreme heat surged by 55%. Unpredictable weather patterns have affected many regions, with extreme rainfall increasing threefold, while the total annual rain has actually decreased. The primary driver of this crisis is the continued reliance on fossil fuels, which contribute over 75% of global greenhouse gas emissions. As these gases blanket the earth, trapping heat, we face rising temperatures and changing climate. Our world is heating up faster than ever before, disrupting nature's balance and putting all life on earth in danger. To bring this issue under control, global carbon dioxide emissions must be reduced by 45% by 2030 compared to the 2010 levels, and we need to reach near net zero emissions by 2050. Carbon capture utilization and storage (CCUS) as a solution to this is facing several challenges due to high capture, transportation, storage costs, lack of a proper regulatory framework, lack of incentives with high upfront investment required for relatively inefficient carbon capture. To face this issue, we at iGEM IIT Bombay are working on an innovative nature-based solution, CalciCapture, to tackle climate change using a unique calcifying green marine algae, Emiliania huxleyi. Through synthetic biology, we are enhancing the calcification process of E. huxleyi, a species of coccolithophore, to capture carbon more efficiently from the atmosphere and convert it into calcium carbonate shells. This algal-based solution sequesters carbon, offering a sustainable approach to combat the climate crisis.
To understand the organism's behavior in its natural environment, we captured both population and cellular dynamics of E. huxley through modeling efforts of the dry lab team. We wanted to get insights about how environmental factors are affecting the net carbon sequestration and what is the bottleneck in the real oceanic setting. Hence, taking inspiration from previous Eulerian phytoplankton models, we did a sophisticated modeling of growth and calcification processes occurring in oceans, particularly North Atlantic, which is a prominent spot of carbon sequestration. The non-living component included nitrates, phosphates, silicates, light irradiance, and temperature, among many other things. The final conclusion of our model is that decreasing the half saturation constant for nitrate can affect the net carbon capture. In fact, it can increase it by a huge percentage. This can be used in media optimization and lab culturing.
Since calcification process within E. huxley is relatively understudied, we decided to model this process right from uptake of calcium ions up to deposition. This process can be broadly divided into three parts: uptake of calcium and bicarbonate ions from the surrounding medium and buffering of calcium ion using the calmodulin protein within cytoplasm, followed by unloading of these calcium ions within the CV, onto which the formation of calcite crystal take place, and finally the deposition of this calcite crystal on the GPA layer onto the cell wall. Since the data was limited, we decided to approach Ria Madan, the co-head of iGEM team within ISAR Mohali, to get insights on how to go ahead. She told us that we should focus on finding out calcification models present within other organisms, wherein we found an interesting paper on bone mineralization. The bone mineralization process takes into account the immature mature collagen forms, the nucleators, the inhibitors, and the calcite crystal formation rate using these differential equations. Also, the process is regulated using this flowchart. We decided to apply this bone mineralization process onto our deposition on the GPA layer, since it is very similar, as GPA is an acidic protein just like collagen. The differential equations were modified in order to take into account our changed system, and the normalized graphs for these were obtained as follows. After this, we decided to move on to coccolith vesicle. The coccolith vesicle interestingly shows that the protein expression of all the proteins taking part within formation of calcite deposition remain unchanged despite the change in rate of calcification. Rather, the whole process is modulated only by pH. The deposition of calcium and bicarbonate takes place onto the a wall of coccolith vesicle, which actively pumps out the proton and maintain alkaline condition within this microenvironment in order to maintain suitable conditions for calcite formation. After this, since we saw that the whole process is modulated only by pH, which is regulated by the concentration gradient. Once the concentration gradient goes below a certain threshold, the CV stops calcification process by decreasing the pH. So acidification of the CV is the only mechanism by which it regulates the overall calcification process. Now we move on to uptake of calcium. The uptake of calcium through the calcium channels present on the membrane is mainly regulated by the concentration gradient and the activation by free calmodulin. Calmodulin, the cytoplasmic protein which buffers the free calcium ions present on the in the cytoplasm, actually activates the calcium channels on the membrane when present in free state, while in bound state the hydrophobic patches get exposed and it stops interacting and activating this process. In order to understand we integrated all the three processes and finally tried to reach to the conclusion. Our conclusions were quite interesting. We found that uptake is limiting over deposition. The blue line here shows the depositional part, whereas the red shows the final part, which almost lies parallel to the calcification based on uptake, showing that uptake is limiting over deposition. Further, uptake was taking into account the amount of free calmodulin and the concentration gradient. In order to find the dependence on these factors, we got the graph plot like this, showing that for 10% increase in the external calcium concentration, the calcification rate increases only by around 2%, whereas increase in expression of free calmodulin by 10% increases the calcification rate by around 11%, showing the importance of expression of calmodulin. Also, increment in external calcium concentration can only go up to a limit, because beyond this limit there is saturation of the calcium channel. This was even correlated with our literature review, which showed differential expression of calmodulin within diploid versus haploid strains of calmodulin.
Now, let us talk about how we figured out calcifying, a protein that we think plays a role in facilitating calcification. We know that Emiliania huxelyi has two stages, a diploid stage that calcifies and a haploid stage that doesn't. As you can see on the right, CAM is differentially expressed in a diploid stage, suggests a link between CAM and calcification. Of course, this is just a correlation, it could mean several things, but it's an important starting point. Interestingly, CAM and huxley has a very similar structure to human CAM, which is well studied. This similarity means we can leverage what we know from the extensive literature on human CAM and apply some of that understanding to huxley as well. Before we dive into the results, let's introduce our powerhouse of analysis, GeneCoder, a tool specifically crafted for future iGEMers to harness the vast amount of publicly available data for gene-level insights. GeneCoder is designed to simplify the complexities of bioinformatics, allowing the users to bypass the steep learning curve of multiple tools and instead focus on actionable insights. With just a few straightforward commands, users can generate ready-to-analyze visualizations thanks to the integration of powerful, proven tools like Callisto, PyTXimport, and PyDseq2, all seamlessly running behind the scenes. While we plan to extend the capabilities in the future, the current version supports three key features. 1. Co-expression analysis, plotting gene abundances, and 3. Differential expression PCA plots. Okay, let's discuss some of the results we found out about CAM over here. The box plot shows the expression of CAM across samples with varied Ca2 plus concentrations, and of course this is thanks to our tool GeneCoder. We observe that CAM expression remains relatively stable until around 1 millimolar of Ca2 plus concentration, after which there is a noticeable increase at 10 millimolar. Interestingly, it's around 0.1 millimolar where calcification begins, suggesting that this site uptick in CAM expression could play a role in initiating the calcification process. Here, we see a clear overexpression of CAM in the calcifying strain compared to the non-calcifying strain. This further reinforces the connection between CAM and calcification. We build over this result again later. Well, we see something remarkable over here. P-trichognathum is the closest calcifying protist to E. huxley, whose CAM sequence we were able to find. This level of structural similarity between these two sequences, with a root mean square deviation of 0.897 and a strong alignment, suggests functional conservation, implying that CAM1 could indeed play a significant role in the calcification process. Now, we move on to human CAM, where we see a high RMSD value of 10.011, a 10x jump from P-trichognathum. This strongly suggested that the functional divergence has facilitated calcification. Finally, here we highlight the transcriptional regulation of CAM1 and Emiliania huxley. As we can see, several transcription factors show a strong correlation with CAM1 in the calcifying strain. However, in the non-calcifying strain, there is little to no correlation. This indicates that CAM1 may be involved in a specific transcriptional network that's active during calcification, and which upregulates CAM1. The conclusion here is that the pattern suggests the CAM1 is a part of a larger transcriptional regulatory pathway in calcifying conditions. Its lack of correlation in the non-calcifying strain hints at its essential role in driving the calcification process.
Despite its immense promise in carbon cycling, E. huxley remains largely underexplored. With its whole genome already sequenced, our goal is to understand its carbon capture metabolic pathways. This is where we come in, building new tools to make a better understanding of the system and unlocks its potential for scalable carbon capture. Through synthetic biology, this is the first time E. huxley is being explored from this perspective, laying the foundation for groundbreaking technologies that could help combat climate change. To date, the recognized carbon capture pathways involves transporter and channel proteins located on the plasma membrane. Our first step is to understand the functionality of one of E. huxley's most important players in calcification, that is calmodulin, or also known as CAM, which regulates calcium ion homeostasis essential for this process. For the same, we talked with Professor Resham Wala, who works with the Center of Energy Biosciences, and discussed and eliminated possible approaches, which concluded to achieve this, we will express and purify CAM in E. coli, establishing a reliable platform for studying the protein outside its native environment. But our vision extends beyond this. Our long-term goal is to engineer E. huxley itself. By developing genetic tools to modify E. huxley at the molecular level, we aim to unlock new ways to enhance its carbon capture capabilities. We selected E. coli as the host organism for E. calmodulin expression because of its efficiency and well-documented application in protein production. Also, based on sequence alignment, it was observed that E. coli calmodulin shows more than 80% similarity to our target protein. To achieve high levels of expression, we could not optimize the calmodulin gene to align with E. coli's proton preferences. Ensuring effective protein synthesis by employing the PET28 vector, which features a T7 promoter, we enabled robust transcription of the calmodulin gene with expression tightly controlled by IPTG induction. Initially, we attempted to insert the codon-optimized gene into the PET28 vector using a restriction deletion method. However, gel electrophoresis revealed that insertion was unsuccessful. This is possible because of double deletion or self-ligation. To overcome this, we shifted our strategy to in-fusion cloning method. We confirmed its success by running the PCR amplified products on the gel where the expected bands were observed and further validated it through Sanger sequencing. Next, we transformed E. coli with this vector and proceeded to express the CAM gene in RIPL and Rosetta cell lines, which is known to show expression of eukaryotic proteins due to presence of additional genes that codes for extra tRNA for some of rare codons. After IPTG induction, we assessed expression level using SGS page, but did not observe a distinct band for calmodulin. To further investigate, we conducted a more sensitive Western blot, which unfortunately also failed to detect calmodulin. So far, we have tested 2 E. coli cell expression hosts and will explore other strains like P. lyse and origami for further purification and characterization of the protein. The protein we are working with has not been previously characterized, making this a novel area of exploration. This may explain the unexpected results during expression, and we are working to address these challenges as we optimize the process.
So as soon as we are able to express and purify our calmodulin, we will perform a series of assays to understand its structure and function. Circular dichroism and fluorescence at 280 nanometer will help analyze how the protein changes structurally in the presence and absence of calcium ions. Assays such as NAD kinase and calcium-specific affinity chromatography can be used to biochemically characterize its function. By optimizing these techniques available for this novel protein, we will be providing tools that others in iGEM community can use to study E. huxley protein in detail. We also plan to identify the interacting partners of calmodulin and develop more understanding of the calcification pathway in E. huxley. We hypothesize that the concentration of CAM is the rate determining step in the calcification, and modulating it could enhance calcification in E. huxley. By confirming that the CAM expression in E. coli is functional and involved in calcification, we aim to gain crucial insight, especially given the lack of data on E. huxley CAM. This will allow us to fine-tune the parameters for the manipulation of E. huxley. To analyze and find approaches to this problem, we talked with Professor Kiran and Professor Balaji and discussed possible attempts. This concluded with the aim to develop genome editing tools for E. huxley using CRISPR-Cas9 homologous recombination. Using genetic modification tool, we would try to modulate the expression level of calmodulin gene inside the E. huxley itself. We have designed a few genome editing tools using the PUC19 vector as a plasmid backbone compatible with the RFC10 standard. We incorporated left and right homology arms for inserting the T7 promoter at the upstream of the calmodulin gene. Also, we would like to target homology regions for the RBCL gene, driven by its promoter to ensure efficient expression of the T7 polymerase and repressive protein. Our work lays a crucial foundation for genome editing in E. huxley, enabling future advancement in the synthetic biology and biotechnological innovations.
While we worked on the technical aspects, our integrated human practices team ensured that the project remained grounded in real-world applications and sought input from stakeholders across various fields to shape our solution. Throughout our journey, we were fortunate enough to receive guidance from academicians and researchers from various universities and institutes in India, helping us find our path in case of roadblocks and giving us valuable technical insights. We contacted Agromorph and C6 Energy, companies that focus on algae-related products. They helped us by giving us information about lab culturing and scaling up, explaining the importance of our work, where it could be used for a number of applications from an industry perspective. Our end product from our project is calcium carbonate in the form of coccoliths, which can be used as a raw material in the cement industry. To learn more about the cement manufacturing process, how carbon capture is currently involved in the industry, at exactly what point in the process do we come in, and what are the things that we should ensure while carrying out this integration, is something we talked extensively with Mr. Jay Prakash Tiwari from Jindal Steel and Power Limited and Mr. Gyanendra Dixit from Ultratech Cement. To gain knowledge about the public perspective on climate change and use of biological solutions to mitigate it, we carried out a survey. Most respondents acknowledged the significant impact of climate change on the world. However, there was a notable lack of awareness around biological solutions for carbon capture and storage.
Building on the insights gained from our interactions with experts and stakeholders through integrated human practices, we also recognize the importance of raising awareness and fostering scientific curiosity. This led us to actively engage in educational outreach, where we aim to share our knowledge by making synthetic biology accessible to a wider audience. Through our work with E. huxley, we are taking meaningful steps towards advancing carbon capture and synthetic biology, contributing to innovative solutions for the climate crisis and a more sustainable future.